So my presentation today will center around a method that myself and computational partners John, Kieran, and Murtash, and Victoria have developed over the past couple of years to enable high-throughput RNA isoform sequencing. The approach is named Multiplexed Array sequencing, or MAS-Seq for short. So traditional gene expression analysis is used to identify genes or transcriptional programs of interest. In the past 5 years or so, the community has really leaned into single-cell RNA sequencing. This really newly enables us to resolve these genetic features at the subpopulation level. Unfortunately, though, this fails to capture the diverse array of RNA isoforms that can arise from any one gene. So these different RNA isoforms derive from alternative splicing, which is a core regulatory process used by cells to diversify the function of genes. And so during the maturation of mRNA, exons and introns are differentially spliced, and this gives rise to mRNAs that differ in their coding sequence and their untranslated regions. Ultimately, this affects the protein isoform that's generated, translation efficiency, stability, localization, and even reading frame changes. And as well, alternative splicing is pervasive. It's estimated to occur in greater than 90% of human genes. And so really beyond the number of protein coding genes, it's this orchestrated transcriptomic diversity that enables the vast biological complexity or phenotypic attributes that we see in complex organisms. And I'm not going to go into it, but there are countless examples of alternative splicing playing essential roles in development and homeostatic balance. As well, when defective, aberrant splicing gives rise to a host of diseases, including cancer, where mutated splice factors are prominent drivers, particularly in the hematopoietic malignancies here. And so again, owing to the central role that alternative splicing plays, genome-wide association studies have identified RNA splicing as a primary link between genetic variation and disease. Splicing quantitative trait loci, or SQTL analyses, have been employed to really discover these genetic variations that impact splicing, either in cis or in trans. And studies have relied on short-read RNA sequencing to measure this percent spliced in of particular exons given different genetic backgrounds. For instance, we can see the difference in a percent spliced in of a particular exon in these genetic backgrounds with a clear trend of this dosage of the T allele going up. Further, allele-specific alternative splicing here provides resolution linking different alleles in heterozygous individuals to the different splicing outcomes. Thus, having the T allele in this gene, you get a higher percentage of percent spliced in. A lot of this work was done on short reads, but as many people are beginning to prove out now, and so certainly hope with MAS-Seq in the future, we'll get greater resolution into SQTL analyses. One final example is, this struck me recently. It's a really profound study from Barbosa et al. Back in 2012. They looked at gene expression and alternative splicing across a number of different species, ranging from frogs to chickens, mice, humans, and other non-human primates. Specifically, they performed RNA-seq on different organs of these species for comparison. So they found that while across organs from different species, the gene expression was much more similar. Though when they looked at alternative splicing, it was actually within the same species, the organs within the same species, alternative splicing was more similar than an organ to a different species. So it has the inverse trend with gene expression, right? And so, this really drives home the evolutionary and regulatory importance of alternative splicing, and also points to some real challenges with model organisms and they're being able to be translated, at least at the splicing level, to humans or to other species. So clearly gene expression analyses are not an accurate reflection of the transcriptomic state of the cell. For example, we have CD45 here, ubiquitously expressed in a population of CD8 T cells. However, when we're able to resolve CD45 isoforms RA and RO, we can clearly see that there are differences in expression. And so we should be mindful that transcript isoforms can have different or even opposing functions, and therefore aggregating these isoforms into their parent gene, this effectively sweeps very meaningful and complex information under the rug. And so, despite this essential role that alternative splicing plays, our ability to resolve these isoforms is very limited. And I have that depicted here in this illustration. We have our prototypical gene, gene X, which gives rise to multiple transcript X isoforms. Traditional short-read sequencing lengths are insufficient to span the multiple splice boundaries within these cDNAs or these RNA isoforms. And thus, it's poorly suited to accurately recapitulate or identify which isoform they came from. Of course, long-read sequencing can sequence the full length of the cDNA, and it can just unambiguously identify the transcript isoform. And so we wanted to quantify this difference between of isoform identification with PacBio or long-read platforms and some of the premier short-read sequencing approaches that boast the ability for some level of isoform resolution. And so we leveraged this synthetic RNA isoform standard from Lexogen called SERVs. And this product is composed of 7 SERV genes, each with their composite set of RNA isoforms. So this is just an example of SERV gene 3 with a number of different constituent isoforms. These are, the sequence composition of these has been tailored to reflect many of the complexities found in the human genome. So for long read, we did PacBio sequencing, which you're all familiar with, and then I performed SmartSeq3 on the SERV library. And briefly, how SmartSeq3 works is you perform first-strand synthesis, creating your cDNA, and at the 5' end, you actually put a UMI sequence. It's in the template switch oligo. You then amplify that sequence through PCR. You then tagment, and then perform paired-end sequencing. And so the sequencing read that goes over the UMI and then the first part of the gene should represent a unique molecule. And then as you're going to be sequencing multiple reads from this parent gene, you're going to have different, because the tagmentation is going to fragment at different areas of the gene, you're going to get different coverage over the read 2 region. You can then, in silico, link and reconstruct these reads, and hopefully the idea is that you'll be able to identify the isoform, right? Yeah, so we can see from this confusion matrix that long reads unambiguously and confidently map to a single isoform from each gene, right? So there's very little confusion. However, this is in contrast to SmartSeq3, which there's low confidence in multi-mapping of these isoforms, of these various isoforms within the genes. There's really just a massive quality difference or capacity to identify isoforms between these two approaches, even with the latest kind of molecular tricks in short read. And this is just an IGV track that highlights the coverage differences between short read and long read sequencing across one of the SERV genes. We can see here that there's always that fixed read 1 that I showed where you get this uniform coverage, and then read 2 coming from this 3' end, the coverage tails and drops precipitously after 1 kb, right? This is in strong contrast to long reads, where beginning to end, right? And so this really highlights what's driving that discrepancy in the ability to confidently identify RNA isoforms. And what's nice is this doesn't come at a cost, right? So MAS-Seq is still highly quantitative and tracks really well with short read sequencing at quantitating genes, and is even more accurate when we're looking at very long molecules. So, while these long read approaches have the read lengths to accurately identify RNA isoforms, they don't yet have the read throughput necessary for many leading edge studies, including single cell and spatial RNA sequencing. And so this figure represents the state of the field. On our x-axis, we have read throughput. On our y-axis, we have read length. And in this dotted box, we have kind of this ideal range for RNA isoform sequencing. Illumina certainly has the read depth, but it really falls short on read length. PacBio and Oxford have the read length, but are limited in their throughput capacity to hit this zone. So in the coming slides, I'll describe how MAS-Seq, which is a modified library preparation method, can boost the throughput of PacBio sequencing and really enable or put researchers in striking distance for many of these high-powered studies. First, I will go in a little bit on how PacBio sequencing works for isoform, for iso-seq. At the top here, we have our cDNA library, of which hairpin adapters are ligated. This effectively circularizes a molecule. From there, it's a strand displacing, it's a sequencing by synthesis reaction using a strand displacing polymerase. The molecule is sequenced, and when the polymerase reaches where sequencing initiated, it will simply displace that strand and continue sequencing in a circular fashion. You get these multiple subpasses that can be consensus based, that can allow consensus base calling, and you get very high accuracy sequencing. That's shown here. On the x-axis, we have our number of passes, and y-axis our Q value, our quality scores. You can see as your number of passes increases, you get this rapid increase in base calling accuracy, but there's an eventual diminishing returns, right? And so, just as like a landmark, after 10 passes, you're sitting around a Q30 score, which is really high quality sequencing. And so, these polymerases will typically traverse 100 to 200 kb, right? And compared to these to this like very large sequencing capacity, RNA isoforms tend to be rather small, around 2 kb or so. And what this results in is this this very excessive amount of passes over these molecules. And so we can see this as wasted sequencing capacity, right? Just kind of incessantly iterating over these molecules. If you look at the quality gains, you're already in massive massive Q score. So no real added benefit. This is in contrast to whole genome sequencing fragments, which are around 20 kb. This is their pass profile, where they get this optimal balance of having enough passes to give you high quality sequencing, but you're also capturing a lot of information. And so in total, we can see isoform sequencing on PacBio as potentially having 18 kb of lost opportunity on that sequencing fragment. Right. So to take better advantage of PacBio, I devised a method to programmatically link cDNAs into these long arrays that match the size of that whole genome, that more efficient whole genome sequencing fragment. And then you can sequence those arrays and informatically deconcatenate them at those landmarks. And then from this, you effectively boost the output of your PacBio sequencing run. So starting from the top, how this works is you take your parent cDNA library that you distribute into multiple parallel PCRs. And then through amplification, you can actually add on these sequential adapters. You'll see here noted in A, B, BC, CD, DE, and so on and so forth. You can then pool, I should note that there are these deoxyuracil bases in the primers, really important. These can be kind of selectively digested away, and this reveals these adapter ends as single-stranded, right? And then they can take place in hybridization with their parent or their partnered adapter sequence. So B prime to B, C prime to C. So after amplification, you pool and clean these amplicons, and then you can perform this deoxyuracil digestion, revealing these single-stranded overhangs. And this will drive very strong and specific hybridization. In the presence of a ligase, these molecules will be covalently bound into one single molecule. And then, as stated before, sequencing occurs, and you get boosted throughput relative to the number of cDNAs that you can concatenate in your array. This is entirely programmable by the user themselves. So notably, this concatenation reaction is very efficient. In red, we have our parent, the size distribution of our parent cDNA library. You can see it's around 1.2 KB. And then after creating a 15x cDNA array, our size distribution is sharply around 18 KB. And then now when we look at our pass analysis, we can see that in blue, our MAS-Seq pass profile strongly resembles that of whole genome sequencing libraries and is in real contrast to the iso-Seq excessive passes. So, we're excited with our ability to efficiently generate these MAS-Seq libraries. The next thing that we wanted to do was apply this for single-cell RNA isoform sequencing. We demonstrated this first by using 10x single-cell gene expression libraries. And so, see, briefly, this system works by encapsulating cells with barcoded oligo decorated beads in a emulsion droplet. From there, cDNA synthesis reaction occurs using those barcoded beads, uniquely labeling those RNAs, associating them at the single cell level. You then have cDNA amplification, eventual fragmentation, and then short read sequencing. There's this very key intermediate where it's the full-length cDNA, right? And so the idea here was for MAS-Seq, we can take this intermediate from the 10x protocol, concatenate them, and then perform long-read RNA isoform sequencing. So we did that, and this is a Sankey diagram breaking down the read metrics from a 10x MAS-Seq run. So we started out with approximately 5 million reads, of which half are CCS corrected. This is like par for the instrument. And then we have this computational workflow that Kieran and John developed called Longbow that can deconcatenate those reads. We had about 2.4 million of those passed filter, and then multiplexing occurred, and we ended up with around 29, almost 30 million reads, and this is about a 12-fold boost in output. And then as well, around just over 2.5 million reads were uncorrected. But the interesting thing is these arrays have those MAS-Seq adapters kind of interspersed throughout the read. And so in these very noisier reads, we're actually able to use those expected landmarks to identify those sequences, and we can actually reclaim those. So we call these reclaimed reads. So we reclaim them, and then we get around another 10 million reads that are typically thrown away. These are of lower base quality, but they're certainly still very usable for mapping and identifying isoforms. Maybe you can't do SNP level analyses with these. Yeah, so diving into the sample that we performed 10x on, this was done in collaboration with Moshe, where they took tumor-infiltrating CD8 T cells from a patient, and then we performed 10x 5' single-cell gene expression. From the same cDNA library, we performed both short-read Illumina sequencing and long-read MAS-Seq. And so these are the UMAP embeddings using either short or long-read sequencing. We can see that at this level there's very high concordance, and we can see the clustering of identifying stem-like memory cells, early activated, leading to early activated exhausted, cytotoxic effectors, and then ultimately terminally differentiated. So we see kind of the whole differentiation gambit of these T cells. And so to demonstrate the added value that MAS-Seq throughput brings, Murtash performed a downsampling analysis going from 33 million reads, which was after filtering kind of the reads that we ended up with for this analysis with MAS-Seq, and then downsampled it all the way down to typical iso-seq levels, which is around 1.6 million reads. We can see that there's a clear kind of evolution of our UMAP, and we can see that we have a 34-fold increase of identified differentially spliced isoforms, going from somewhere around 47 to north of 1,600. So really, really large gains with regards to both clustering and identification of differentially spliced isoforms. Next, we took advantage of the canonical splicing patterns of CD45 in these immune cell subsets. And so CD45 is like heavily used by immunologists to identify the different different states of T cells. And so I'll quickly review them. There's the splicing occurs mainly in this region. There's an A exon, B exon, and C exon right next to one another. So this would be called CD45 ABC. Then there's any permutation of these exons, so CD45 AB, A, BC, B, and if there are none of these exons, it's CD45 O, I should say RO, so RA, RO, etc. And so we performed site-seq, which is a simultaneous single-cell gene expression, but also protein expression analyses. You can order these antibodies that are specific either to CD45 total, CD45 RA specific antibodies, and then RO specific antibodies. And they have this oligo that you sequence, and that's a proxy for the presence of that protein. So this is how you get protein expression through single-cell analysis. And so when we look at CD45 total protein expression in our single-cell data, we can see that it's, again, ubiquitous across the population. When we look at the RA and RO antibody protein expression, we can see that there's clearly differences. RA is more on the naive side of our populations. RO is on the more differentiated side. We're really excited to see that MAS-Seq recapitulates these findings at the RNA level. So on the right-hand side of the embedding, you can see that the RO isoforms are clearly expressed on the right side, strong concordance with the site-seq. And then this is where it really gets interesting. We have our CD45 RA expression, again, mostly on this left-hand side. When we look at our A-containing isoforms for MAS-Seq, we actually can discriminate much better. There's CD45 RAB and RABC. And we can see that there are different patterns of expression within the subpopulations, right? This protein expression is based off of an antibody discriminating the A epitope. It doesn't have any capacity to understand what's going on elsewhere in that protein, right? And so this is, we're actually much better able to discriminate the different isoforms here at the RNA level. And then we're also able to identify CD45-RB and RBC, for which we didn't have antibodies for, right? So, and then again, there's clearly differences in expression across these B-containing CD45 isoforms. So, I think it's worth repeating that MAS-Seq does not require specialized antibodies or reagents, and it's able to discriminate between closely related RNA isoforms. So it should be a powerful tool for isoform discovery. And finally, we leveraged pseudotime analysis to resolve CD45 isoform progression, splicing progression, and expression of its associated splicing factor. So we visualized our single-cell data in pseudotime, and so on this force-directed graph, we can see our cell populations going from stem-like memory to early activated to, let's see, early exhausted and then terminally differentiated. And this is showing that in pseudotime. And then here we can see the splicing of our CD45 protein or gene, starting at ABC in the most naive or earliest in pseudotime, going down. And we can see this monotonic increase in expression of HNR and PLL, its known splicing factor. So this is really exciting because, of course, we did this in a known case, but you can imagine taking any data that you have, if there's any relevant pseudotime trajectories in your data, you can look for certain transcripts that undergo some sort of patterning in their differentiation, and then also see if there are kind of associated increases in expression of any splicing factors, and this can give you, putative hits for splicing interactions. Yeah, so now with MAS-Seq, we can go beyond gene expression and actually measure transcript isoforms. This is the actual nucleic acid information that's being translated into proteins. We're hopeful that this fundamental advance will catalyze this transition from gene to transcript level analyses and have broad impacts across many fields of biology. For a more technical summary, we get around 35 to 40 million reads per flow cell at current chemistries. We expect this to go up as we refine MAS-Seq itself and as advancements get made on PacBio side, on their platforms. This is really well poised to co-evolve with the platform. It's compatible with most protocols to generate full-length cDNA, including existing 10x single-cell libraries. You don't need to do anything special. If you have 10x libraries hanging out in your freezer, follow the protocol, and you can get MAS-Seq data on them. And this does certainly depend on your samples. People are doing wild things with 10x these days with super loading, but if you're doing regular loading, a single run can be sufficient to provide statistical power and embedding and clustering for and identify differentially spliced isoforms. And something I'm just very excited to announce is that the Broad, specifically the genomics platform and the data sciences platform, are planning to productionize MAS-Seq, and this is targeted for the second half of 2022. You'll be able to submit cDNAs for MAS-Seq library construction, sequencing, and data processing for the following library types. And so more information will follow as we begin to roll that out. And shown here is just a sampling of how we're looking to apply MAS-Seq at the Broad Institute. Clearly, very excited for alternative splicing in both the context of development and disease, GWAS studies, proteogenomics, neoantigen identification, many fun things to do on the functional genomics side, and of course, transcriptome reference generation. Yeah, so at the top, I just have to thank John, Kieran, Martash, and Victoria for leading this work with me. To the Cohen and Blaney labs, Nir and Paul in particular, for their excellent guidance and support during the development of this project, and all the many other contributors that made this work possible. Thanks to the many members of the genomics platform for working with us to support this new run type and bring productionization to MAS-Seq. And then, of course, our sources of funding, most notably the Spark Award, which really catalyzed taking this from a concept to proof of principle. So I'll take any questions.